active transport is where we are today. Active transport means moving things into and out of the cell using the cell's energy. Meaning the cell has to be an active participant in moving these things into and out of the cell. When we looked at those pictures of A, B, and C, diffusion, osmosis, and facilitated diffusion, the lipids, the proteins, they didn't do anything. They just sat there. The molecules moved themselves. In active transport, we're talking about things that are either too big to even get through with facilitated diffusion, so we really have to work at getting them into and out of the cell, or we're talking about moving the molecules in a direction that they don't want to go in, like bailing out a ship, bucketing water out of a ship. The water wants to go into the ship if you got a leak, but do you want the water in your ship? No. Like with the contraction thing, that would be an example of active transport. The water moving into the cell was passive, but when they squeezed that contractile vacuole, that was active. All right? So let's talk about energy first, because it requires energy to do active transport, so you need to know a little bit about energy. We briefly mentioned energy when we talked about the molecules in the beginning of the year. Do you remember which two of those macromolecules dealt with energy? Carbohydrates was one. Lipids was the other. In those molecules, energy, the stuff that we were talking about, is stored in the bonds that hold the atoms together. If you think back to lipids or carbs, and if you can remember what the pictures looked like, there were these huge chains, especially in lipids, of carbon atoms bonded together, usually surrounded by some hydrogens or something. The reason carbohydrates and lipids are so good at storing energy is because they have tons and tons and tons of bonds. And the more bonds you have, the more energy you can store. When you want to release that energy, like if you eat some carbohydrates and you want to get the energy that they have inside of them, you have to break the bonds. You have to pull those atoms apart. If you eat a chocolate bar or a pixie stick, those carbohydrates are just sitting there unless you actually break them apart, and then you can get the energy that you have stored in each and every one of those bonds. That's why we have to digest our food, and that's why once the food gets to our cells, they have to break it down even farther. That's the mitochondria's job. Remember we said that mitochondria takes the food and breaks it down and makes energy. Well, this is what they're doing. They're taking the food and they're breaking those molecules apart. And when we said that they were making energy, what we really meant was that they were making ATP. ATP is called adenosine triphosphate. What does tri stand for? Three phosphate molecules. And that's why we call it ATP, because nobody wants to walk around saying adenosine triphosphate all the time. ATP is a molecule that provides easy-to-access energy. In your reading yesterday, what did they compare ATP to? Something dealing with money. There you go. Your carbs and your lipids are like a $100 bill. $100 bills are awesome. Hey, that's $100. But if you go to the store, do a lot of stores take $100 bills? Gotta be careful. You can go to the mall maybe and spend it, but if you go to like the food court or something, they're gonna look at you like you were from another world. And especially if you try to go to like a vending machine or something like that, it's just gonna spit it right back out at you. It can't handle that much. ATP is like little bitty $1 bills. You can spend those anywhere. And if you have 100 of them, you have just as much energy as you had, or just as much money in that $100 bill. You just have it in bills that are easier to spend. It looks kind of weird to walk around with $101 bills, but you know, it's better than having that big $100 bill if you're going someplace where you can't spend it. I like to think about this in terms of food, because food, you think about storing energy in food, like carbs, like candy bars or something like that. I like to think about carbs and lipids as a great, huge, big old pot of gumbo, which it's cold enough outside that is actually kind of appealing. Mom makes big old pot, like we're talking about like monster pot of gumbo in the beginning of the week. Do you eat all that at one time? No. What do you do with the stuff that you're not going to eat? You put it in Tupperwares. And if you want people to be able to help themselves, what kind of Tupperwares are you going to put it into? You're going to put it into small Tupperwares. Like if you want people to be able to bring it for lunch, you want to put it in little single-sized Tupperwares. To me, that carb or that lipid is a huge big old pot of gumbo, whereas the ATP is the little Tupperware. You have, and I can't fit Tupperware, that's what that says. You have enough energy in one molecule of glucose, which is a what? Carb or lipid? It's a carb. It's the monomers of sugar. Like if you digest sugar, that's what you get, is glucose molecules. You have enough energy in one molecule of glucose to make 36 ATPs. 36 individual single serving sizes packets of energy, which is what ATP is, in just one glucose molecule. That's why your body makes ATP. You don't always need 36 single serving packets of energy. Unless you've got 36 people to feed, are you going to eat all 36 things at one time? If you were serving gumbo? I don't think so. You know, you can make a big party and invite everybody over. That would work. But if you're just doing this for your family, you're not going to eat 36 servings at one time. When your cell gets some glucose from the food that you eat, it's not going to use all that at one time. Instead, it wants to break it down and store it in these little bitty packages so that when it needs some energy, it can use as much as it needs. If you needed $5, you could use $5 bills and you'd be able to do that. But if you had a whole entire $100 bill, you may not be able to use it. If you needed 5 units of energy, you could use 5 ATPs. If you tried to use a whole big glucose, you'd be wasting 30-something things of energy. You wouldn't need it. The energy in an ATP molecule is stored in the bonds that hold the phosphates together. When we look at the picture, I'll explain that one again. We said energy is in the bonds that hold molecules together. So for ATP, the energy that we're getting out of it is holding the phosphates together. This is what ATP looks like. I'm going to scratch off 2 of these P's real quick. Now what do I have? This is on your exam. You have adenine and ribose and a phosphate group. Not a protein. What is adenine part of? DNA and RNA. This is a nucleotide. It's not a DNA nucleotide because this is just ribose. So what is it? It's an RNA nucleotide. It's got adenine and a sugar and a phosphate group. That's a nucleotide. ATP is nothing more than a nucleotide with a couple extra phosphates attached to it. Phosphates are not friendly with one another. I think that would be a easy statement to make. Phosphates really don't like to be attached to one another. So it takes a lot of energy to hold them together. And in ATP, the energy that we're talking about storing is stored right here in the bonds that hold those two phosphates together, those three phosphates, triphosphate. When you want some energy to do something in your cell, you take an ATP molecule and you break off one of the phosphates. And you release the energy that was stored in holding that phosphate onto the molecule. One of the great things about ATP is that they're reusable, just like Tupperwares. Once you use the energy, you can clean it out, put something else into it. Oops, went the wrong way. And make ATP again. You can use the energy from a new molecule of food and put that P right back on. And when you need it somewhere in the cell, you break it back off. On, off. It requires energy to make it. You release that energy when you break it. And that's what that little formula up here at the top means. ATP has 3 phosphates, T for 3. ADP, diphosphate, has how many phosphates? 2. This is a DP plus a P for phosphate. And what we can really do with this equation is put another arrow right here. That means, oops, that's supposed to be an arrow. My penmanship today is just not good. That means that I can take ATP and break it apart and make ADP and an extra phosphate and release the energy. Or I can put it back together again and make ATP. I can go back and forth as much as I want to. Over and over and over again. ADP is when you take it off, because there's only 2 phosphates. ATP is when you put it back together again, which I keep, because that has 3 phosphates. All right, let's talk about what we're going to use this energy for. We're going to use this energy to do a lot of things in the cell. One of those would be active transport. There are 2 types of active transport. There is bulk transport and there are protein pumps. Have you ever heard of bulk shopping? Like if you go to Sam's? Tell me what that means. You buy a lot. You don't just get one little container of peanut butter. You get like a 4-pack of peanut butter, and that's all you can get when you go to Sam's. They have huge packages of things. That's bulk transport. That's bulk buying. It means you buy in big supplies. Bulk transport, therefore, is going to mean we're going to move what? Really big things into and out of the cell. Endo means into. So endocytosis is moving really big things into the cell. Endocytosis is moving really big things, and we're talking about too big to even fit through facilitated diffusion into the cell. For instance, we've actually studied this once before. Do you remember our little amoeba and how he ate? He hugged his food to death. Here was his food. He surrounded it with his little pseudopods, and he said, I love you, I love you. Oh, come here. Let me give you a hug. Now you're my lunch. We've already mentioned endocytosis. I just didn't use that word. What word did we use instead? We used phagocytosis. Phagocytosis means taking in solid particles. So things like food, really big food. Anytime you see PHAG, that means food or eat, one of the two. We've talked about macrophages. When we talked about the immune system, we said that those were big eaters. And how do they eat? Well, they eat using phagocytosis, just like anything else does. So when it sees a little bacteria, it says, oh, look, lunch. And this is how it works. It stretches out its little pseudopods, its little extensions. Am I right? It doesn't always do it anymore. All right. And it surrounds the food until the food is actually inside the cell. and then it can digest it. Pinocytosis is the same thing, except it's taking in liquids. Pinocytosis, P-I-N-O, means taking in liquids. We're not talking about water here. Water can move through the membrane. It fits. We're talking about really thick, syrupy, oily liquids, things that aren't going to fit through the molecules because they're just too thick and viscous to do that. With phagocytosis and pinocytosis, really the cell isn't paying attention to what it's eating, it's just eating it. What's the problem with that? Yeah. You've got these little guys who are hanging out on the outside of the cell going, oh look, if I get in here, I can make babies. And if the cell's not paying attention to what it's taking inside, it could take inside a virus or a bacteria or something bad. So there is a third type of endocytosis. It's called receptor-mediated endocytosis. Receptor-mediated endocytosis identifies the object using proteins in the membrane before it takes it in. Well, it has to be something that the cell recognizes, or else it won't take it in. Receptor-mediated endocytosis identifies the object using proteins in the cell's membrane before it takes it in. Sometimes the viruses can disguise themselves, and in that case, receptor-mediated, even though it's trying to protect itself, doesn't always work. The viruses that looked like this, the one that were in the video, they could actually inject their DNA themselves, but there are some, like the circle ones, that don't have that ability. Instead, they pretend to be food and let the cell take them in. They just don't get digested. Then possibly, yeah. Yes, ma'am. You could say moving really big objects if you wanted to. If endo means moving things into, what is exo? Moving things out of. Exocytosis is moving really big things out of the cell. Yeah, we're not going to go into different ones. We've got enough new words today. Really, you don't ever talk about the different types, not in a normal biology book, but they do talk about phagocytosis versus pinocytosis a lot. It could be waste. It could be a protein that the cell produced that needs to go somewhere else. It could be a hormone that the cell makes that needs to go to another cell, like your ovaries when they produce estrogen. The cells are making that estrogen and the cells release it, and this is how they release it because the molecule is just too big to fit through any other way. Or when your neurons talk to one another, which is what we're going to study Thursday. All right. Bulk transport aside, there is another type of active transport, and that's using a protein pump. I have two pictures up here in front of me. One is a picture that we have seen before or have seen something like. What is going on in this picture? That's facilitated diffusion. Tell me what a facilitated diffusion protein looks like. It's like a tube or a tunnel or I don't know what else you want to call it. It's open on both ends like a paper towel roll. And it lets things just go back and forth as much as they want to. And it can, if the cell wants it to. And some channel proteins stay closed until they're needed, and some are just open all the time. It just depends on the channel protein. These facilitated diffusion proteins have a name. They're called channel proteins. And I remember them because I think channel kind of looks like tunnel in the spelling. So underneath channel protein, you have a place to put a visual description. So we said that it looks like a tube. It's open on both ends. Does it actually do anything once it's open? It just sits back and lets the molecules move themselves. So does this require energy? No. And if you want to put an explanation, you can say the molecules move themselves. That's why it doesn't require energy. Now, protein pumps are used for active transport. And protein pumps, if you look at the picture, they don't look the same, do they? Tell me about a protein pump. They have one end that's open and one end that's closed. It's kind of like making a little V with your fingers. Yes, ma'am. Say it again. The dots are any type of solute particle that we're talking about. So in facilitated diffusion, the molecules are moving themselves, and they can just go through and back and forth as much as they want to, because the channel protein is open on both ends. With a protein pump, the molecules can't go all the way through, because the protein pump is always going to be closed on one side. And it requires the protein to move or to rock, which is what that protein is doing, like this with your fingers. You can put your fingers together and leave your bottom parts open and then make a V and just rock back and forth. That's what a protein pump does, because it's only wanting to move certain molecules through. Yes. Only certain ones can fit inside the protein. Well, it requires energy in order to make the molecule move back and forth. So what are we going to need? The ATP molecule. We're going to need an ATP in order for this to work. And typically with a protein pump, we're not moving the molecules in a direction that they actually want to move. That's why this is called active transport. Let's look at it a little bit more up close and personal. Protein pumps are always closed on one side, and they rock back and forth. They're always going to be closed on one end. When they rock back and forth and back and forth and back and forth. They're always going to be closed on one side. Their job is to move things in a direction that they don't want to go. They're moving things against the concentration gradient. They're moving things from where there's not so many to where there's lots. And that's backwards from what we have been talking about. Molecules, we said, they don't want to be crowded together. And that's why this requires energy, because you're moving the molecules in a way that they don't want to be moved. I'll go back to my example of bailing out a boat. If you've got a leak in your boat, what's the water going to do? It's going to fill that leak. It's going to come into the boat, spread itself out in all this nice empty space, and sink your boat, unfortunately. So what are you going to do? You're going to either plug the hole, or if you can't do that right away, you're going to take a bucket and you're going to fling that water back out of the boat. You're going to move the water from where there's not so much yet to where there's already lots and lots and lots in the ocean or the lake or wherever you're sitting. And the water's trying to go in. It's like your little paramecium. The water was coming in, and what did he do? Squeezed it back out. We're moving the molecules in a direction that they don't want to go. And that's going to require energy to make them go that way. You're forcing them. Does it take energy to climb up a slide? If you're going up the wrong way? All kids do that. Why do kids do that when there's a perfectly good ladder? But they climb up the slide anyway. And that requires a lot of energy. Where does sliding down the slide require energy? No, that's the way you're supposed to go. So we're moving things opposite of what they need to do. And it requires energy because we're moving things against diffusion. Mm-hmm.
Is this like the same thing as the other one? I don't see like where is it trying to get? Is it going to the same place where it becomes going, or is it going out?
Well, it depends on what molecule you're talking about. For instance, salt or sodium is a molecule that your cells really just don't want inside. And we said that the membrane was designed to keep it out. Well, if you do happen to have some inside, you would use a protein pump to pump it back out.
So it takes it from the inside and pushes outside.
Mm-hmm. And then there are some things that your cell really, really need to use. Even though it already has some inside, it can never get enough. And so it would use active transport to pull more into the cell, depending on what we're talking about. That's what protein pumps are for. It has to be something small enough to fit through that protein, so we're not talking about anything really big. That would be bulk transport.
How does it like get it? If it's going into the thing, obviously it wants to go down, and then it's letting it go down.
Let me show you the animation. Let me get this one last dot out the way, and then I'll show you an example. An example of a protein pump is called the sodium-potassium pump. The sodium-potassium pump pushes sodium or salt out of the cell, and it pulls potassium into the cell. Protein pumps are usually named after whatever molecule they're moving. In this case, we're moving sodium and potassium at the same time. Normally, they only move one thing. Like there's a calcium pump that your muscle cells use in order to make your muscles contract. And it's only moving calcium. Sodium, potassium, for some reason, moves at the same time. You have